Hello students, today we will discuss two important things related with urea cycle and pyrimidine nucleotide synthesis. One is connection between carbamyl phosphate synthetase 1 and 2, and second important thing is why there is orotic acid excretion in type 2 urea cycle disorders. So coming to the first, carbamyl phosphate synthetase 1, okay, CPS 1 and CPS 2. See, carbamyl phosphate synthetase 1 belongs to urea cycle. It belongs to urea cycle, while CPS 2 it is pyrimidine nucleotide synthesis. It catalyzes the condensation of CO2 and ammonium ions. CO2 and ammonium ions. CPS2 causes the condensation of CO2 plus glutamine. CO2 plus glutamine. The N-acetyl glutamate is required. It is an allosteric activator. It is an allosteric activator of CPS1, carbamyl phosphate synthetase 1. Here, the N-acetyl glutamate is not required. This CPS2, carbamyl phosphate synthetase 2, it is activated by phosphoribosyl pyrophosphate. Is activated by phosphoribosyl pyrophosphate. CPS1 urea cycle, first two reactions of urea cycle occurs in the mitochondria. So it is mitochondrial enzyme. Mitochondria. It is cytosolic. Pyrimidine synthesis occurs in the cytosol. And this CPS2, it is under feedback inhibition by the cytidine triphosphate, which causes feedback inhibition of this CPS2. Now, what is the reaction? CPS1 catalyzes the condensation of CO2 plus ammonium ions, condenses, 2 ATPs are required. N-acetyl glutamate is the allosteric activator of carbamyl phosphate synthetase 1 and lead to the synthesis of carbamyl phosphate. This is the reaction catalyzed by the CPS1. This reaction belongs to urea cycle. Now coming to the second reaction, that is means the pyrimidine nucleotide synthesis. It will start from CO2 plus glutamine. The condensation reaction synthetase, the synthetase belongs to ligase, requires ATP. So 2 ATPs are required in this reaction catalyzed by carbamyl phosphate synthetase 2 and synthesis of carbamyl phosphate occur. Carbamyl phosphate occurs. Now this is the first, that is difference between CPS1 and CPS2. Now the second point in our discussion is why there is orotic aciduria. Orotic aciduria or orotate excretion in type 2 hyperammonemia. Why there is orotic aciduria in type 2 hyperammonemia. Now what is type 2 hyperammonemia? It is a urea cycle disorder due to defect in see, ornithine is converted into citrulline in the urea cycle, and this reaction is catalyzed by ornithine transcarbamylase. This is again mitochondrial enzyme. This is again mitochondrial enzyme. So this is type 2 hyperammonemia. Type 2 hyperammonemia. So ammonia level increases. This belongs to urea cycle disorder type 2 hyperammonemia due to defect in ornithine transcarbamylase enzyme. Now why there is orotic aciduria in this disease? Whenever there is defect in ornithine transcarbamylase, whenever there is defect in this enzyme, there is accumulation of carbamyl phosphate. There is accumulation of carbamyl phosphate, and this carbamyl phosphate, it is diverted for pyrimidine nucleotide synthesis. It is diverted for pyrimidine nucleotide synthesis. Now see this reaction, carbamyl phosphate. It is an intermediate in the pyrimidine nucleotide synthesis. Okay, carbamyl phosphate. In the pyrimidine nucleotide synthesis, this carbamyl phosphate, it is converted into. So how will you remember the pyrimidine nucleotide synthesis? CAD, 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 O2, O2. These are the enzymes of pyrimidine nucleotide synthesis. CPS2, aspartate transcarbamylase, dihydroorotase, dihydroorotate dehydrogenase, OPRtase, and OMP decarboxylase. Decarboxylase. So these are the enzymes of CAD-DO2. So these are the enzymes of pyrimidine nucleotide synthesis. Now this carbamyl phosphate, this is the second. It is acted upon by the aspartate transcarbamylase, and there is condensation with aspartate to form carbamyl aspartate. Carbamyl aspartate. This carbamyl aspartate, it is converted into third. This is first, this is second, this is third with the help of dihydroorotase. With the help of dihydroorotase, it is converted into dihydroorotate. Orotate. Now this dihydroorotate, it is converted into fourth enzyme with the help of dihydroorotate dehydrogenase. Fourth enzyme, dihydroorotate dehydrogenase, it is converted into orotate. It is converted into orotate or orotic acid. So here the NAD is required, which is converted into NADH. Okay. Now this orotate with the help of fifth enzyme, OPRtase, that is orotate phosphoribosyl transferase. OPRtase, orotate phosphoribosyl transferase, it is converted into OMP, orotidine monophosphate. So PRPP is required, phosphoribosyl pyrophosphate is required, which provides the ribose. Okay, so it is PRPP is required for this reaction. It is converted into OMP, and with the help of OMP decarboxylase, orotidine monophosphate decarboxylase, it is converted into UMP, uridine monophosphate, and this through various steps. UMP is converted into UTP, UTP is converted into cytidine triphosphate. Through various steps, it is converted into thymidine monophosphate. So this is pyrimidine nucleotide synthesis. Now the important reaction, see the important reaction, OPRtase and OMP decarboxylase. These are the two enzymes. If there is defect in these two enzymes, that causes the orotic aciduria. So it is related with the pyrimidine nucleotide metabolism. Defect in these O2, OPRtase, that is orotate phosphoribosyl transferase, and OMP decarboxylase, that causes the orotic aciduria. Now our point of discussion is why hyperammonemia type 2 causes orotic aciduria. When there is defect in the ornithine transcarbamylase, there is accumulation of carbamyl phosphate. It is diverted for pyrimidine nucleotide synthesis. Now, in the pyrimidine nucleotide synthesis, this carbamyl phosphate is converted into orotate. This carbamyl phosphate, it is converted into orotate. And this orotate is excreted in urine, that causes the orotic aciduria, because excess of carbamyl phosphate is accumulated, which is diverted for the orotate synthesis, and orotate excretion in urine occurs. And these are the two important causes of orotic aciduria, that is OP-ARTase and OMP decarboxylase deficiency. Got the point? Why there is defect? Why there is orotic aciduria in type 2 urea cycle disorder? Because of ornithine transcarbamylase deficiency, carbamyl phosphate is diverted for the synthesis of orotate, and orotate excretion occurs in the urine. So this is a reason why orotic acid excretion occurs in urine in type 2 hyperammonemia. So keep watching. Thank you.